Welcome back to the deep dive. And if you are tuning in right now, I'm going to make a pretty safe bet.
Oh, yeah.
You are likely sitting at a desk, surrounded by this fortress of printed PDFs, maybe a cold cup of coffee, and you have that very specific knot in your stomach.
The one that only a looming regulatory exam can produce.
That's the one.
It is a universal experience. I have to memorize 50 acronyms by tomorrow morning. Panic. I know it well.
Exactly. And we know that when you're staring at a stack of guidelines, the words just start to swim together. Safety, efficacy, quality. It all blends into this gray soup. So today, our mission is to pull one specific high-stakes document out of that stack and make it stick. We're doing a focused deep dive into the ICH-S9 guideline.
Specifically, the non-clinical evaluation for anti-cancer pharmaceuticals. This is the Step 4 version, harmonized way back in October 2009.
That's the one. And looking at this, it doesn't feel like just another safety guideline. I feel like if standard drug development is a calm, orderly drive down the highway, obeying the speed limit, checking your mirrors, S9 is, well, it's an ambulance ride.
That is actually a perfect analogy. It's an ambulance ride.
Right.
The rules of the road completely change when the lights and sirens are on.
So usually when we talk about safety guidelines, like the famous ICH M3, which covers general pharmaceuticals, the philosophy is do no harm. It's extremely risk-averse.
Absolutely.
You have to prove a drug is incredibly safe in animals before you even dream of giving it to a human. But S9 seems to flip that script entirely.
It does. And to understand why, and honestly, to pass your exam, you have to get your head around the core philosophy.
Okay.
In standard drug development, the enemy is potential toxicity. We are terrified of side effects. But in the context of S9, the enemy isn't toxicity. The enemy is time.
Because of the patients involved.
Precisely. This guideline applies to pharmaceuticals intended for advanced cancer. We are talking about serious, life-threatening malignancies where, current therapies have failed or they simply don't exist. These are patients facing a fatal prognosis.
So the whole risk-benefit analysis just shifts completely.
Completely. I mean, think about it. In a standard trial for, say, a seasonal allergy pill, a serious side effect like liver damage is unacceptable, full stop.
Of course.
But if a patient has 6 months to live, they and the regulators are willing to accept significant toxicity if there's a chance, even a small one, of extending survival. S9 is designed to cut the red tape that doesn't actually add value for these specific patients, so we can get potential cures into the clinic faster.
Okay, so time is the enemy. That's our anchor point. That makes sense. Now, before we get into the nitty-gritty rules, and I know there is some tricky math coming up that everyone gets wrong, we need to define the playing field.
Yes. The scope.
You mentioned advanced cancer, but exams love to trick you with exclusions. What is S9 not for?
This is where I see students lose points all the time on multiple choice questions. S9 is very, very specific. It does not cover cancer prevention.
Oh.
So if you are developing a drug to prevent breast cancer in healthy women who might be at risk, you cannot use S9. You have to use the standard, rigorous M3 guideline.
That makes sense. Prevention implies the person isn't sick yet, so you just can't justify those risks.
Exactly. S9 also excludes the treatment of symptoms or side effects. So an anti-nausea drug for chemotherapy patients, not S9.
That's a supportive care drug.
Right. And finally, it excludes things like vaccines and cell or gene therapies. Those have their own very complex rules.
What about healthy volunteers? I know in phase I trials, we often use healthy college students to test safety and pharmacokinetics.
That is the biggest trap of all. If your clinical trial design uses healthy volunteers, you absolutely cannot use S9. You must follow ICH M3.
Why is that such a hard line?
Well, just think about the ethics. S9 allows for toxicity that causes, tissue damage, organ toxicity, maybe even DNA damage. You cannot ethically justify inflicting that on a healthy volunteer who gets no benefit from the drug.
Right.
S9 is strictly for treating sick patients who need the drug to survive. If you are testing on healthy people, you have to play by the do no harm rules.
Okay, got it. If the subject is healthy, S9 is off the table. Now let's talk about the goal of these studies. In a normal toxicology deep dive, we'd be talking about the NOAEL, the no observed adverse effect level. That's usually the holy grail, isn't it?
It is. Finding the dose where absolutely nothing bad happens.
The clean slate.
In the standard world, yes. But in S9, if you run a study and find a dose where nothing happens, you have effectively failed.
You failed.
You probably haven't dosed high enough. In oncology, we are not looking for safe. We're looking for tolerable. We need to find the MTD, the maximum tolerated dose.
And to find that.
And to find that, we need to identify the DLT, the dose-limiting toxicity.
So we are explicitly hunting for the moment the body starts to break down.
We are depending on it. To kill a tumor, you usually have to push the patient right up to the physiological limit. We need to know where that limit is. We are looking for the edge of the cliff, not the safe meadow far, far away from it.
That is a huge shift in mindset. And I imagine that shift just ripples through the actual testing requirements. We've categorized the big changes into what we're calling the S9 exceptions.
Yeah, let's break these down.
Okay, so concept number 1, duration. I read in the source material about a 3-month rule. How does that work?
This is a massive, massive efficiency driver. Imagine you're developing a drug for diabetes or hypertension. Patients will take that drug for decades.
For life, yeah.
So before you can market it, you need to run 6-month, 9-month, sometimes even 12-month toxicology studies in animals to see what happens with that long-term chronic exposure.
Right, because if I'm taking a pill for 20 years, you need to know what it does to my liver over time.
Exactly. But for advanced cancer, sadly, the life expectancy is often short. The priority is immediate treatment. S9 states that for marketing supporting phase 3 and registration, a 3-month repeat dose toxicity study is usually sufficient.
Wait, so even if the drug is successful and the patient takes it for a year, you only need 3 months of animal data?
Generally, yes. I mean, there are rare exceptions, but for the most part, 3 months is the cap. That saves huge amounts of time and resources. You don't have to wait a year for a chronic study to finish before you can file for approval.
And what about recovery? In standard toxicology, if you damage a rat's liver, you have to wait and prove it heals completely, right?
Usually, yes. You have a recovery group of animals that you leave alone for a few weeks to see if they get better. But S9 says the demonstration of complete recovery is not essential.
Not essential.
You just need to show that recovery is possible or predictable based on the science.
Predictable recovery. What does that mean in practice?
It means if you damage the bone marrow, which, many cancer drugs do, we already know bone marrow regenerates, you don't necessarily need to wait around to prove it reached 100% baseline again. As long as the trend is moving the right way or the mechanism is understood, you can move forward. Again, it's about saving time.
Okay, moving to concept number 2, safety pharmacology. Usually this is a whole battery of separate tests, checking if the heart stops, if the lungs keep working.
Right. The famous HERG assays and respiratory studies. In standard development, those are often standalone protocols requiring their own sets of animals. S9 says, stop wasting time in animals. Do not do them separately.
So we don't check the heart.
No, we do check, but we incorporate it. If you're running a toxicology study in a dog or a monkey, just hook them up to an ECG during that study. Collect the data while you're already dosing them for general toxicity. Don't run a separate experiment just for safety pharm. It's about consolidation.
Okay, that makes sense. Efficiency again. Now, concept number 3. This is the one that really shocked me. The don't do it list. S9 basically says entire categories of standard testing are just optional.
Or not needed at all. It's not that they are optional, it's that they are considered irrelevant to the patient's immediate crisis. Let's look at fertility. Standard drugs require extensive fertility testing. S9 says, not needed.
Because the patient is fighting for their life.
Exactly. If a patient is facing stage 4 cancer, the potential risk of reduced fertility in the future is secondary to the immediate goal of staying alive. Often the patients are older, or the treatment itself is known to be toxic to reproductive organs anyway.
So we don't need a study to tell us that.
Right. Same goes for pre- and postnatal toxicology. We generally don't run those studies.
What about genotoxicity? Checking if the drug damages DNA.
This one's a bit nuanced. For clinical trials to actually start testing in humans, you don't need genotoxicity data. For marketing, you do need the data, but here's the reality. Most cancer drugs work by damaging DNA. They are designed to kill rapidly dividing cells by shattering their genetic material.
So the drug is genotoxic by design.
Often, yes. So you run the test, it comes back positive, and the regulator says, okay, noted. It doesn't stop the drug, it's expected.
And the big one, carcinogenicity. Testing if the drug causes cancer. This feels like the ultimate irony.
It is the irony born of necessity. Carcinogenicity studies take 2 years to run in rodents. 2 years. The life expectancy of the target patient population is often less than 2 years.
Right.
The risk that a drug might cause a secondary cancer 5 years down the road is, academically interesting, but clinically irrelevant if the patient doesn't survive the next 6 months.
So we just skip the cancer-causing tests for cancer drugs.
Generally, yes. S9 says carcinogenicity studies are not needed to market an anti-cancer drug for advanced disease.
Wow. Okay, one exception I saw in the notes, though, embryo-fetal toxicity.
Yes. This is the one you cannot skip for marketing. We need to know if the drug causes birth defects.
Because a patient with cancer could be pregnant.
Exactly. We need to be able to tell that patient this drug will likely harm the fetus. They need that information to make an informed decision. But, and here's the S9 speed kicker, you don't need that data to start the clinical trials. You only need it later for marketing.
It really is all about removing barriers to entry.
Yeah.
Okay, that is a lot of do this, don't do that.
Yeah.
You mentioned you had a way to keep this straight in our heads for the exam.
I do.
Yeah.
If you are sitting in that exam hall and your mind goes blank, just remember to ACT.
ACT, walk us through it.
A is for advanced cancer only. Remember the scope. It's not for prevention, not for healthy volunteers. It's for the serious stuff.
Okay. A for advanced.
C is for cut the extras. No fertility, no standalone safety farm, no carcinogenicity. Cut the fat.
And T.
T is for tolerance. The goal is the MTD maximum tolerated dose. We are looking for tolerance, not safety.
Advanced cancer, cut the extras, tolerance, ACT. That is actually really helpful. I can see that being a lifesaver on a written test.
It organizes the chaos.
Okay, let's shift gears. We've covered the philosophy, we've covered the exclusions. Now, we have to do the math.
The calculation of the starting dose.
This is the part where everyone gets nervous. We are about to give this experimental drug to a human being for the first time. How do we decide the dose? I assume we don't just, guess.
Definitely not. Note 2 of the guideline gives us a very specific formula. And it's tricky because the formula changes depending on whether your data comes from a rodent, like a rat, or a non-rodent, like a dog or a monkey.
Okay, let's visualize this. I have a rat study. I have my toxicity data. What's the number I'm looking for?
For rodents, you are looking for the STD-10.
Acronym alert. Break that down for me.
Severely toxic dose in 10% of animals. So you look at your data and find the dose that caused severe toxicity, but not death in 10% of the rats. It's a fairly high bar of toxicity.
Okay, I have that number. What do I do with it?
You take that dose and you divide it by 10. That is your starting dose for humans.
110th of the STD-10.
That's it.
Okay. Rodents STD-10 divided by 10. Easy enough to remember. 10 and 10. Now, what if my data is from a dog, a non-rodent?
The rules change. See, rodents are less predictive of human response, so we use a harsh safety factor on a severe endpoint, but non-rodents are usually more predictive of human biology.
Okay.
So we look for a different number.
The HNSTD.
Which stands for?
Highest non-severely toxic dose. This is a much safer baseline. It's the highest dose where we saw no lethality and no life-threatening toxicity. It's basically the ceiling of safe.
So for the rat, we look for a toxicity marker, the STD-10. For the dog, we were looking for the safe marker, the HNSTD.
Exactly. And because it's a safer starting point to begin with and the animal is more predictive, we don't divide by 10, we divide by 6.
16th of the HNSTD.
Correct. So to recap for the test, rodents get the severe endpoint, STD-10, and a 110th cut. Non-rodents get the safe endpoint, HNSTD, and a 16th cut. That distinction is subtle, but I guarantee that's a multiple choice question. Which factor do you use for a dog study? If you say 110th, you're wrong.
You're wrong, and you might underdose the patient, wasting valuable time, or if you use the wrong endpoint, you might overdose them. It's critical math.
Right. Now, while we are on practical application, there's one more scenario. Combination drugs. This is standard in oncology now, cocktail therapy. Drug A plus drug B.
It is the standard of care. Rarely do we treat with just one agent anymore.
So my gut instinct says mixing chemicals is dangerous. 1 + 1 might equal 5 in terms of toxicity. I probably need to run a whole new toxicology study for the mixture, right?
Your gut instinct is noble, but it would fail the S9 exam.
Of course it would.
The guideline is very strong on the 3R principles: reduce, refine, replace. We want to minimize animal use. S9 says, if drug A is approved and drug B is approved or well characterized, you generally do not need a new toxicology study for the combination.
Really, we just trust that they won't, explode when mixed.
Well, unless there is a specific pharmacological reason to suspect a massive synergistic toxicity, like they target the exact same pathway in a dangerous way, yes, We rely on the individual data.
Yeah.
The rationale is that we can monitor for overlapping toxicities carefully in the clinic.
Okay.
We don't need to sacrifice more animals just to prove that two toxic drugs are, in fact, toxic together.
That's a key point. It's not just about speed for humans, it's about ethics for animals, too. The 3 R's are baked into the guideline.
They are. It prevents a lot of unnecessary testing.
Okay, we have covered a massive amount of ground. We've gone from the philosophy of time is the enemy to the calculus of 16th HNSTD. Before we let our listeners go, let's condense this.
Good idea.
If they walk into the exam room and their mind goes blank, what are the essential takeaways?
Let's boil it down to the big 6. If you remember nothing else, remember these.
Hit me. Takeaway number 1.
Scope. S9 is for advanced cancer only. It excludes healthy volunteers, prevention, and symptom management. If the patient isn't fighting for their life, S9 doesn't apply.
Takeaway number 2.
The goal. We are looking for the MTD, the maximum tolerated dose, not the NOAEL. We expect and accept toxicity.
Takeaway number 3. The math.
Start dose. For rodents, it's 110th of the STD10. For non-rodents, it's 16th of the HNSTD. Do not mix up your denominators or your acronyms.
Takeaway number 4.
Duration. A 3-month toxicity study is usually sufficient to support marketing. You generally don't need chronic studies.
Takeaway number 5.
The don't do it list.
Yeah.
No fertility studies, no standalone safety pharmacology, and generally no carcinogenicity studies to market the drug.
And finally, takeaway number 6.
The 3 R's, S9 pushes to reduce animal use. No unnecessary combination studies. Trust the data you already have.
There you have it. The cheat sheet for ICH S9.
Before we sign off, I want to leave the listener with one final, slightly provocative thought. We've spent this whole time talking about how S9 cuts requirements, how it removes hurdles.
Right. Cut the extras.
But there is a section on immunotoxicity that sort of hints at the future. Usually, S9 says standard tox studies are enough to check the immune system. But if you are developing an immunomodulatory drug...
Which, let's face it, is where all the modern cancer breakthroughs are coming from.
Exactly. Checkpoint inhibitors, small molecules that tweak the T cells. S9 suggests that for those drugs, standard testing might not be enough. You might need extra endpoints, like flow cytometry or specific biomarker analysis.
So while the guideline's about streamlining, it's not about being lazy.
Precisely. It reminds us that cutting red tape doesn't mean ignoring the mechanism of action. If your drug works by tweaking the immune system, you better be watching the immune system closely, rules or no rules. The science always wins.
That is a great perspective. It keeps the patient and the science right at the center of it all.
Indeed. And for those of you doing the reading, do take a look at Table 1 in the document. It lays out the dosing schedules, how a weekly clinical dose translates to an animal schedule. It's a great little reference.
Excellent advice. Well, to all the learners out there, good luck with the exam. You've got the logic, you've got the math, and you've got the ACT mnemonic. You're going to crush it.
Trust the logic of the guideline. It's all about risk management to save lives.
Thanks for listening to this deep dive. We'll catch you on the next one.